So next up, we'll have Jason Kwan, who's here at UW-Madison. He'll be talking a little bit about metagenomics, which is one of the sources of genomes that Eric touched on earlier today. So I'll turn it over to Jason.
All right, great. All right, first of all, is the volume okay? You guys can, yeah, all right, great. Okay, so yeah, first of all, thank you to Christina and Myron for inviting me to speak to you today. This is a really great opportunity for me to talk about my research. And as this title slide says, I'm going to talk about metagenomics and small molecule biosynthesis. And I guess, this subject came up earlier in the questions after Eric's talk, but a logical place to start is what is, oh, hang on, it's not good. Oh, yeah, okay. Logical place to start then is what is metagenomics? And I guess I'm going to take a bit of a step back here and just tell you about the microbiome, which is what we study in metagenomics. So it's now known that all living things on Earth, or all of the ones we've studied so far, have a microbiome associated with them. So, in other words, a complex community of bacteria that are specific to the little environments on or within that organism. So, we have a microbiome, animals have a microbiome, all mammals, insects, and so forth. And these are populations that are different from the bacteria you find just in the environment. And even the environment itself has a microbiome. And different environments have different microbiomes. So, for example, different depths of soil or different surfaces under the sea, hot springs versus seeps in the ocean and that sort of thing. And but the fundamental problem with studying microbiomes is a very small portion of these bacteria are readily culturable in the lab. So estimates vary, but it's probably something around 0.1% or even lower of the species that are out there we can easily grow in the lab. So how do we study this microbiome that contains all these unculturable things or mostly unculturable things? Well, it turns out it's actually pretty easy to homogenize an entire sample, so an entire beetle or the gut contents of a human or or a swab from skin or something like that, and just extract all of the DNA from all the biological material within that sample. And what you end up when you do that, what you end up with when you do that is a huge sort of complex mixture of lots of DNA from lots of different organisms that originated from the sample itself. Now that poses a few data analysis problems. The first of which is that when you start doing this sort of thing, you don't necessarily know how many species are there present in the sample in this huge mixture. A lot of the species could be unknown to science at that point, so we don't even know what their genomes should look like or anything about them at all. And the other problem we have in metagenomics is that we're essentially randomly sequencing. This is called shotgun sequencing. And if you want to look at rare species, well, their DNA is also going to be rare in that mixture. And that means you have to sequence really, really deeply in order to see these rare species. So I'm just going to give you one example of how we've used metagenomics. Here is a beetle. It's called Legria villosa. That's its scientific name. And this is a soybean pest that is found in Brazil. So it kind of predates soybean plants. And the way that this beetle reproduces is it lays its eggs in these little bundles and it sort of buries them in the soil. And I guess that hides these from predators, but it also has a disadvantage, which is that the soil has a lot of microbes in it, it has its own microbiome. And there are lots of species of fungi in the soil that can easily infect these eggs and prevent them from developing properly into beetles. So in other words, if they get infected by fungi, they're less likely to develop into adult beetles. And so you can imagine that there is an evolutionary pressure here to develop some way to prevent infection by fungi of these eggs. And my collaborators, Martin Kaltenpoth and Christian Hertweg, found out that this is actually the case. There is a compound that can be isolated from the eggs called legriamide. And this is an antifungal compound. And they were able to show through ecological experiments that presence of this compound reduces the infection frequency of these eggs. And so therefore, when this compound is present, more adult beetles can survive. Okay, so you may be wondering, where does computation come into this area of study? And the issue here is that we have a huge volume of data we have to get through. So there are different methods of sequencing. And I'm not going to go into huge detail about this, but in general, there are two types of sequencing that have different trade-offs. You can either sequence really, really deeply, but you get lots and lots of very tiny sequences called reads. Or you can have another type of sequencing where you get longer reads, which give you more information, I suppose, but it's more difficult to sequence really deeply. Now, the problem here is in metagenomics, we have this huge complex mixture, and so we really need to sequence very, very deeply. So often we have to resort to the type of sequencing where you get lots of short reads and a huge volume of data. And this is called Illumina sequencing after the company that makes those sequences. And so what we're looking at here is the type of data. This is the raw sort of data we get. The reads themselves are very, very short, about 150 base pairs long. And if you think about the size of a genome, so a bacterial genome is maybe 5 megabases, human genome is about 30 billion base pairs. This is just a very, very tiny fraction of that. But you get hundreds of millions of these reads, so that kind of makes up for this. And what this means, I guess, in terms of the data size, is your data can be up to about 100 gigabytes or more, just the raw gzipped data you get from the sequencing machine. And the fact that these are very short reads means that you have to process these in order to put together long genomic sequences to figure out what your genome actually, or what your metagenome and genomes look like. And the way that you do this is through a process called assembly. And somewhat counterintuitively, it turns out that the best way to make assemblies is actually to split up these very short reads into even shorter fragments called k-mers. And at that point, you make a De Broglie graph. Hopefully, I'm pronouncing this correctly. I don't really say it out loud that often. This graph consists of edges, which are the k-mers themselves, and the nodes are k-1 fragments that different k-mers share, right? So neighboring k-mers overlap by k-1. And this huge graph is constructed for all of the reads in the sample. And through that, you can follow the graph around to reconstruct the original sequences. And you can see in this example that you can even handle repeats under some circumstances. So in this case, it's going rounds and looping rounds. That's showing a repeat to reconstruct this original sequence. And in practical terms, we use k-mers, we use multiple different values of k-mers between about 33 and 120. And this sort of is how we reconstruct the genome. Now, I am at roots a biologist who has sort of learned computing along the way. So these assembly algorithms are kind of above my pay grade, but I will just say that this is a very interesting area of computer science. And if anyone is interested in this, these algorithms still need to be optimized. The primary assemblers that we use are programmed by other people. So we use metaspades and megahits. And the problem here really is that, of course, this complexity scales really sort of drastically as soon as you get graphs as your graphs sort of increase in numbers of nodes and edges and so forth. And remember that we start off with hundreds of millions of reads, and we're splitting them up into like, k equals 33 sections, and that's just a huge amount of data. And so this requires a huge amount of memory. And it scales not just with the data size, but also the complexity, right? It depends on how many unique different k-mers you have in your data and the proportion of those k-mers. And in practical terms, what we found is that with megahit, we can assemble most metagenomes in under 1 terabyte of RAM. but it doesn't give as good results as metaspades. But metaspades itself takes up a lot more time and memory. There doesn't seem to be an upper memory limit. We've used, we actually sort of partnered with a company called Memverge to try and assemble some very complex soil metagenomes. And they gave us a machine that had, they have these special machines which use SSDs in place of RAM. And we were given one that had 3 terabytes of this sort of SSD RAM type thing. And our job didn't finish in 1 month. So these things can take weeks. So MetaHit is okay results, I suppose, but and it takes a couple of days. Metaspades can take up to a couple of weeks. And I think at some point we did have a special arrangement where we could use with CHTC, we could use the high memory nodes and had permission to run these jobs for like 2 weeks at a time. I'm not totally sure if we have that still because we haven't done one of these in a while. Okay, so that's assembly. That's the first challenge here in this field. The second is after you've got an assembly, well, you've now got a huge mixture of sequences that are slightly longer than reads, but they're still sort of all jumbled up, and we don't know which sequence belongs to which species, and we don't even know how many species we have. So the assembly itself can be quite big because remember, this is not just one genome, but lots and lots of genomes from the organisms that were in the sample. And our job then is to sort of sort these out into bins where each bin is part of a genome from a single species. So this is a process called binning. And this is often what we want to do in order to find out who is doing what in a metagenome. And so there are a number of programs that do this sort of job of binning. They all suffer from a certain issue, which is that they separate out the contigs, that's what these longer assembled sequences are called, based on things like nucleotide composition or coverage. And while these are different for different species within a metagenome, they can also be the same, right? There's no reason why things can't have the same nucleotide composition and they can't have the same coverage. So we actually designed our own computer program to use more orthogonal sources of information that can also be inferred from metagenome. And we call this autometa. So how this works is we start off with our assembly that I showed you before. And the first step is to simplify this by looking at where each of these sequences come from. So in other words, we're working out if a sequence is from a bacterium or it's from a eukaryote or it's from a virus and so forth. And how we do this is we essentially just look at all of the protein coding genes within these contigs. So we first have a gene finding search where we find where all the coding sequences are. We take those protein sequences and we basically blast them against public databases, and then from that infer what the taxonomic group of that sequence, where it came from. And this can take a lot of computation because, with a huge metagenome, we can have up to about 1 million genes. So if you think about it, that's 1 million blast searches, each time searching against all the entire database of every sequence that's known. Recently, this has become difficult because as the database has got larger, but we've started using a more simplified database to make this a bit quicker. And this is one of the tasks where CHTC really comes in handy because we can just submit, those million jobs and figure out our different bins here. Now, this is useful, of course, because say, if we're only interested in bacteria, we don't want eukaryotic sequences contaminating our bacteria. And lots of binning programs don't do this step. And so you end up with these weird bins which have both bacteria and eukaryotes in them. So after that, we concentrate on the fraction we're interested in, say it's bacteria. And we then do a step that's lots of other binning pipelines do, which is examine the frequency of k-mers within each of these sequences. So this is where k equals something short, like 5 base pairs or so. And we can put that through a sort of dimension reduction step, for example, Barnhart stochastic neighbor embedding or BHTC, to make these types of plots where the points are sequences and the distance between the points are roughly proportional to how different they are in terms of nucleotide composition. And you can see that the species kind of form discrete clumps. We then put it through a sort of clustering algorithm, and this is where the computer basically figures out what groups are and what are likely the different bins. And at this point, we do something that's a bit different to other binning pipelines, which is that we try to assess the completeness and purity of the bins that we get. And we do this by looking at specific genes that are known to be present as single copies only in bacteria. And the way that we do this is not through blast searching, but through another type of searching called hidden Markov models. And I'm not going to go into the details because I don't think I'm really qualified to say what hidden Markov models are. But I will just say that the way I think about them is that they are very more specific types of blast searches where you're concentrating just on the residues that confer function. So what we're doing here is we're looking for specific genes and we have a set of about 140 of them. And what we see is how many of them we see, which tells us the completeness, and how many of them we see with more than one copy in a group. And that tells us how much contamination is likely to be present. We then siphon off the sort of good bins based on some criteria. So for example, more than 90% complete and pure. And we're left with some unbinned fraction. And what we can do, actually, I should mention here that we don't just cluster with one set of parameters. We do a sort of parameter sweep here and maximize for the greatest number of bins over this criteria. The unbinned things, we can then sort of put through this process again and maximize the number of good sort of bins we get out. And we can do that over and over again until we don't get any more good bins out. And so this helps basically sort of figure out, well, the reason behind this is because there's no one set of clustering parameters that will give us all of the good bins. There's a combination of different ones. And another thing we do even before this process is to sort of split this data up even further, because the less sort of complex your data is, the more clustering is effective. And so, for example, here we have all of bacteria. We could split that into different phyla. So, example, phylum Proteobacteria, and then do this process, and then have another phylum and do this again, and so forth. So, just to summarize, we use CHTC for a lot of the sort of steps in Automata. For example, well, but even before that, doing our assemblies, which require a lot of RAM, up to a few terabytes sometimes. We do our large-scale sort of blast searches and our large-scale HMM searches. Some of the problems we come across, well, assemblies are really, really demanding. As well as the RAM, you need very fast sort of data access because there's lots of reads and writes that go on. And network storage is really not fast enough. It really sort of extends the times of the jobs. So in addition to the amount of RAM we need, we also probably need about that amount or maybe double of very fast local access disk as well. And at present, Automata is based on a sort of Nextflow pipeline. It would be nice to be able to sort of integrate that into Conda some way. For now, we sort of manually sort of do the different steps ourselves. So I think we're out of time, so I'm not going to go through this other sort of example I was going to talk about, but I will just, I'll just put this acknowledgement slide up so I can make sure I thank all the people that are involved in this. So all of the names in red are the people that were involved in the Automata project, or the other thing that I didn't have time to talk about. So thank you, everyone. I'll take questions if there are any.
All right, questions for Jason. You did have like 1 or 2 more minutes.
Oh, okay. I could go on for a long time.
Questions? Oh, we've got one in the back.
So if you'll let me ask a question about something I know nothing about. On the assembly pieces where you need enormous amounts of RAM, that kind of reminds me when I talk to some of the climate scientists where they say, for simulations, we could always use faster, they need a infinitely fast computer with zero latency and an infinite amount of memory, and then they will be happy. For assemblies like that, is there anything that you can do stochastically where you partition the problem, take some good enough, or maybe distribute out lots of good enough solutions at the smaller scale and then combine? Is there a stochastic approach or something that doesn't require just
Right.
almost brute forcing the graph, which
Well,
in the end you will always want more RAM, right?
the closest I've come, well, so okay, that's one reason why I brought this up, is because I think in this room, maybe at least someone is going to be interested in this, and it's kind of beyond me. But as far as I know, like no one's really figured out a good way of parallelizing and splitting up the problem, because essentially what you need to do is like figure out, where all the k-means are and how the graph works. And I don't really know how you split the graph up ahead of time. The closest I've got is that, of course, when you have a huge metagenome, sometimes there are a number of species that are really high coverage. So what you can do is you can actually just assemble 1/10 of the data and get those very high coverage things, and then subtract those from the reads and redo your assembly with everything. And I've done that a few times, and that sort of works okay. That's probably the most I've been able to come up with. There probably is some kind of computer science answer, so that's one reason I kind of brought this up. If anyone has any ideas, yeah, let me know.
Yeah, certainly. From a throughput computing standpoint, the more that we could do, I don't have that many 6 terabyte machines.
Yeah,
but
well,
we can
exactly.
give
This is the
you
problem, right?
dozens of 0.5 terabyte machines, right?
Yeah.
We have an online question from Taylor Lee of Edgewood, and the question, which I don't understand, but maybe you will, is if you had every almost genome, would binning replace the metagenome assembly?
Sorry, can you repeat?
If you had every almost genome, would binning replace metagenome assembly?
Would binning replace?
Metagenome assembly.
Oh, well, yes. Some people, a lot of people approach me at conferences and say, "Oh, well, your job is not going to last long, right? Until long read sequencing, replaces all of this." But I think it's very optimistic to think that long read sequencing will always give you reads that encompass entire chromosomes. That's never going to happen 100% of the time. So I think there's always going to be a need to figure out what parts of genomes belong to other parts. Is my take on this.